So in the next video I'm just going to tell that how this insulin and glucagon are going to play their role. Till now what we have understood, if I just summarize the previous video in one line, that would be that insulin causes dephosphorylation, glucagon causes phosphorylation. Okay, now another concept that you need to know that I understood in the previous video was that insulin is a predominant hormone in the feeding state and glucagon is a predominant hormone is a fasting state. Okay, and since in feeding state when insulin is the predominant hormone, so insulin will predominantly cause all the dephosphorylation of the enzymes and will activate them. On the other hand, it will cause inactivation of three enzymes after dephosphorylation. That would be two enzymes of glycogenolysis, that are glycogen phosphorylase kinase, and third enzyme would be hormone sensitive lipase. And glucagon in fasting state will cause the inactivation of all those enzymes which are activated by dephosphorylation by insulin, but it will cause activation of three enzymes which are inactivated by dephosphorylation of them by insulin. So now I'm going towards carbohydrate metabolism, and I'm going towards carbohydrate metabolism in case of feeding state, and fasting state. But remember this thing, before going towards that what happens in a combination way, you have to understand two main things. One is linked to carbohydrate mechanism, other is linked to lipid metabolism, and these two steps and their regulation are very important to understand before starting full-fledged feed fast cycle. So this step you should understand, and this step is very important concept. If you see, this is your fructose 6-phosphate and this is your fructose 1,6-bisphosphate. Fructose 6-phosphates convert into fructose 1-phosphate. This step going in this direction is a step of glycolysis, and going in a reverse direction is the step of gluconeogenesis. Okay, in gluconeogenesis you make glucose from your non-carbohydrate sources. So this steps got reversed because ultimately after reversing this, fructose 6-phosphate will form a glucose 6-phosphate, and glucose 6-phosphate, after the action of glucose 6-phosphatase, this will be converted into glucose. Okay, so but what is important is that fructose 6-phosphate and fructose 1,6-bisphosphate is this step is irreversible. This step is a reversible step. But and you should also understand the concept that glycolysis and gluconeogenesis, they cannot occur simultaneously. Any one of them can occur. Any one of them, these processes can occur at a time. In feeding state, since we have enormous amount of glucose, we need to store the glucose. We do not need to form glucose. So glycolysis is the predominant step which we need. Okay, glycolysis is the predominant step in case of feeding, and gluconeogenesis is the predominant phase in the fasting, because gluconeogenesis releases glucose in the blood. We do not need it because we are not in a fasting state right now, we are in a feeding state. So what you need to remember is that glycolysis it occurs in your feeding state, whereas gluconeogenesis is the predominant phase in your fasting state. Okay, this concept you should know, that glycolysis predominantly occurs in your feeding state and gluconeogenesis occurs in your fasting state. Clear this thing. Okay, so this step, fructose 6-phosphate, will go into fructose 1,6-bisphosphate in your feeding state, and this reverse step occurs in your fasting state. Now how does it occur? Okay, how does it is regulated? You can clearly see that there are two enzymes, PFK1 and FBP1. In fact, these are two activities of the same enzyme. Okay, this is phosphofructokinase which causes this to convert into this, and this is phosphatase which causes this to this. It will cause the loss of one phosphate and convert into fructose 6-phosphate. So clearly these are the two activities, kinase and phosphatase. These are the activities of two enzymes. Now which will be active in what state is determined by a regulation mechanism. And what is that regulation mechanisms is this. You can clearly see here this is written that is a single enzyme, and this enzyme is PFK2 FBP2. Okay, this is PFK2 FBP2. What we just look upon is PFK1 and FBP1, but this is actually PFK2 and FBP2. Again, it is phosphofructokinase 2 and fructose based phosphatase 2. Now this is a single enzyme, but it has two properties like the one PFK1 FBP1. Okay, and the two properties this enzyme has is one is kinase, that is of PFK2, and other is phosphatase, that is of FBP2. Okay, basically this is a single enzyme, but at one time it acts like a kinase PFK2, and other time it acts like a phosphatase. Now what is important, that is important, you need to understand that when this enzyme, okay, when this enzyme is dephosphorylated, okay, when this enzyme is dephosphorylated, now you can understand which enzyme will cause its dephosphorylation. Insulin will cause dephosphorylation of this enzyme. This enzyme, when dephosphorylated, will be having, after dephosphorylation, it will be having predominantly the kinase activity. And this is what is written here. Dephosphorylation of the enzyme favors kinase activity, and phosphorylation of this enzyme favors phosphatase activity. Means this enzyme will be predominantly a PFK2 after dephosphorylation, and it will be predominantly of FBP2 after phosphorylation. Now who will cause dephosphorylation? Insulin. And when will insulin cause its effect? In the feeding state. So in feeding state, I can say that kinase is active in this enzyme, and in fasting state phosphatase is active. Okay, so after activation of kinase, what happens? This kinase, it will cause fructose 6 phosphate to convert into fructose 2,6 bisphosphate, and phosphatase will cause fructose 2,6 bisphosphate to convert into fructose 6 phosphate. It will look like similar, but these are not similar. You remember this thing, this is 2, and that's why here it is 2. Here it was PFK1 and FBP1. It is 1. These are different. These enzymes are different from what I am just telling. So when in feeding state insulin is active and it causes dephosphorylation, kinase is more active. The predominant substrate that is present is fructose 2,6 bisphosphate. And similarly, in case of glucagon dominated fasting phase, fructose 6 phosphate is the predominant thing. Now once you understand this concept, you should know another thing, that is the fructose 2,6 bisphosphate is the allosteric activator of PFK1, meaning that in this enzyme PFK1 is allosterically activated by fructose 2,6 bisphosphate. You need to cram this thing right now. So when fructose 2,6 bisphosphate would be more, PFK1 would be more active and will cause this step to go into fructose 1,6 bisphosphate. Fructose 6 phosphate to go into fructose 1,6 bisphosphate. And that is why, if you understand again, you will see that when kinase is active, fructose 2,6 bisphosphate is produced more. So fructose 2,6 bisphosphate will cause activation of PFK1, and which will cause fructose 6 phosphate to convert into fructose 1,6 bisphosphate. The opposite occurs during the glucagon dominated phase. When phosphatase is active, more is fructose 6 phosphate that is produced, and less amount of fructose 2,6 bisphosphate is present in the around the cell, and this will cause a relative inhibition of this PFK1. So since PFK1 is inhibited because fructose 2,6 bisphosphate is reduced, okay, so fructose PFK1 will not act, and the step will be reversed. Fructose 1,6 bisphosphate will be converted into fructose 6 phosphate due to predominant fructose bisphosphatase of 1 activity. So that is what I was telling, that in this is how the mechanism acts, that in case of feeding state, insulin dominated phase, glycolysis is favored in fed state, gluconeogenesis is favored in fasting state. And that is why glycolysis occurs in feeding state, because insulin causes phosphorylation of PFK2 FBP2 single enzyme with converting it to predominantly kinase, which will cause activation of fructose 2,6 bisphosphate from fructose 6 phosphate, and this fructose 2,6 bisphosphate will be acting over FEFK1 because it's the allosteric activator of this enzyme, and will causing the enzyme to this step going in this direction glycolysis, and the reverse occurs in this. Now another concept that is important in understanding the fast feed cycle after that glycolysis gluconeogenesis concept is that you need to understand the two main mechanisms. See, there are two steps, two processes: fatty acid synthesis and fatty acid oxidation. You can correlate with these two processes with gluconeogenesis and glycolysis, because one was using glycolysis was using to produce ATP, whereas gluconeogenesis consumes ATP. Similarly, here also fatty acid synthesis consumes ATP and fatty acid oxidation uses ATP because it breaks down the fatty acid to produce energy. And you clearly can see that in one process fatty acid oxidation, fatty acids are broken down to produce energy, and in another process fatty acids are synthesized. So obviously these two processes cannot occur simultaneously, like similarly, which we studied just now, that gluconeogenesis and glycolysis cannot also occur simultaneously, and one process has to occur, so there must be a regulation like in the glycolysis gluconeogenesis part, and that is what regulation concept I'm going to tell you. Look, this enzyme you can clearly see, which is written here, this acetyl CoA carboxylase ACC. This enzyme is the first step of fatty acid synthesis pathway, and it is a rate limiting enzyme of fatty acid synthesis pathway. What is this step causes? This step causes the acetyl CoA to convert into malonyl CoA. Okay, you can see it, acetyl CoA to convert into malonyl CoA. And when it causes this, okay, this causes this acetyl CoA to convert into malonyl CoA, you should understand the concept that this is the fatty acid synthesis that is occurring. But why fatty acid synthesis occurs? Because this enzyme, I just said that insulin causes dephosphorylation, it causes the activation of enzyme, except three enzymes which it makes inactive after dephosphorylation. And since acetyl CoA carboxylase is not among those three exceptions, so after dephosphorylation it will be becoming active, and after phosphorylation it will be becoming inactive. So you can clearly see that in feeding state, where insulin's predominant hormone that is acting, it will cause dephosphorylation of this enzyme to make it active, and in fasting state, where when glucagon is predominant enzyme, it will cause its phosphorylation to make it inactive. So this means that acetyl CoA carboxylase becomes active in case of your feeding state, and it will produce more and more malonyl CoA. Okay, more and more malonyl CoA. Now, what is important thing to understand is that you need to see here that malonyl CoA, it inhibits CPT1. Now, what is CPT1? CPT1 is carnitine palmitoyl transferase 1 enzyme. Now remember this thing, CPT1 has a role to take part in fatty acid oxidation. It has a role to take part in fatty acid oxidation. CPT1 is an enzyme of fatty acid oxidation. It helps in entry of fatty acids into the mitochondria for fatty acid oxidation. So since you know that acetyl CoA is converting into malonyl CoA, and malonyl CoA almost put brakes on CPT1, so it is ultimately stopping fatty acid oxidation. This means that clearly fatty acid synthesis if it is fatty acid synthesis that is predominantly active, it will ultimately cause the inhibition of fatty acid oxidation, and that is why the two processes cannot occur simultaneously. Now, when the patient is in fasting state, or the person is in fasting state, acetyl CoA carboxylase enzyme would be inactive because glucagon will cause its phosphorylation. And after phosphorylation, when it will become inactive, this acetyl CoA carboxylase will not be active, and malonyl CoA levels will be down, and CPT1 will become active because the brakes on it will be released, and CPT1 will be active, and the predominant pathway which will be active then in fasting phase is fatty acid oxidation.